against DNA polymerase E1 in tuberculosis. My name is RagaVishagan. I am a junior at Cupertino High School and a biochemistry student at Foothill College. And this project was done with the University of Missouri. Dr. Kamal Singh is my mentor, and I work with Dr. Eiichi, who's a medicinal chemist. Yes, Dr. Eiichi, Dr. Amy has 2 FDA-approved drugs. Background. So, tuberculosis is a major global concern today, as it is one of the top 10 causes of death in the world. According to World Health Organization, it kills around 10 million people per. It infects about 10 million people per year and kills around 1.3 million. There are treatments available, but around 16% of people who are treated with the current treatments develop drug resistance, and this leads to many strains of multi-drug resistance or extensively drug-resistant strains of tuberculosis. All of these drugs act on cell wall synthesis or RNA transcription, so there is a need for a novel approach in finding drugs that have activity against tuberculosis. For this, we chose to target the DNA E1 polymerase of tuberculosis. This was done because by inhibiting this polymerase, it is fatal to tuberculosis. There are 2 main ways to inhibit this molecule. The first is to use nucleoside analogs such as RIA, which act as chain terminators, or by using molecules similar to nargansine, which was developed by Merck, which intercalates at the DNA enzyme interface and blocks replication. For this project, we chose to find molecules similar to nargansine, as RIA is often excised by the PHP domain of DNA E1, rendering it ineffective against tuberculosis. Our approach was to search through a large ligand library, such as the Chem library with 2.5 million compounds, for a molecule that works similar to nargansine. First, we wanted to figure out which code that we could use for docking. So we settled on RDoc-Vina, but first we wanted to see if it could replicate the results that nargansine had in the lab. So first, we scored the experimental position of nargansine in DNA E1, and we got a Vina score of negative 9.5, which corresponds to a high binding affinity, as it is very negative. Then we redocked nargansine into DNA E1 to see what position it would dock in, and it found it in a position that was extremely close to the experimental position, and gave a Vina score of negative 10.4, indicating that Vina was able to accurately reproduce results seen in the lab, and this validated our approach of using Vina to dock these ligands. The next step was to dock 2.5 million compounds. To do this, we developed a pipeline called the SPDoc drug generating platform. This platform has 3 main modules. The first module filters molecules based on drug likeness to reduce the amount of total time required for docking. The second step performs a high throughput docking, and the last step gets the data from the docking and shows the top compounds. For our first step of filtering, we filtered based on these conditions. These conditions were chosen to reduce the number of ligands from 2.5 million compounds down to 500,000 compounds, which would take a lot less time to dock. Like, the 2.5 million compounds would have taken 500 days at a rate of 200 compounds per hour to finish docking. But 500,000 compounds would only take 100 days. But this is still not ideal. That is where the next step of our platform comes in. This step uses the Swarm workflow cluster in the University of Missouri supercomputer to parallelize the process of docking to complete the docking of 500,000 ligands in only 55 minutes. This is a major breakthrough, as we are able to increase the speed of docking by many orders of magnitude. The top compounds from the initial docking were shown with the final module of our program, and these molecules unfortunately do not show many drug-like characteristics, but we believe that fragments from them could be used to create higher binding affinity compounds. For example, our top compound from this search of the Chemical library only resulted in a Vina score of negative 8.9, which is less than that of nargansine. But we used Vesmifra to fragment these molecules to isolate fragments that would hopefully have a high binding affinity, and we hope to recombine them into molecules that would create a high, that would be better binding. The top 100 compounds from the previous ritual screening were taken and then fragmented and recombined to 1,000 new molecules that we then docked again using the SPDoc pipeline. The Vena scores from this docking that were better, the molecules with Vena scores better than negative 10.0 were chosen. Here is the top molecule structure. Unfortunately, I cannot show you the full structure as it is still pending patent. This structure has three main problems in it. One is that it contains a substructure of morphine, which is not ideal for a tuberculosis drug. The second problem is that it has a spiro-attached barbituric group. A barbituric group has high, shows central nervous system suppressant properties, and a spiro attachment is very hard to synthesize in the lab, rendering this molecule somewhat useless. But we were able to use a three-step scaffold hopping approach to change this molecule into a more suitable form. For the first step, we wanted to change morphine. However, our initial attempts at changing morphine were not very successful, as greatly altering the structure caused it to lose its binding affinity. Here's one example of a successful change of structure that was able to keep the binding affinity, but was still not significant enough for us to be comfortable in using the structure. In the structure, I added a singular oxygen to cause a reinforcing to change the structure, but it wasn't good enough. But these new structures were not completely useless because by superimposing their best binding positions, we were able to see where certain hydrogen bond donors and functional groups had to be for a high binding affinity. This led us to discovering this other substructure, which was able to maintain the same binding affinity whilst removing morphine, allowing us to solve one of the three problems with this molecule. In the next step, we wanted to replace barbiturate with a different group that didn't have central nervous system suppressant properties. We used a Python probe to replace it with many different fragments. And here's an example of how a certain substructure would have looked like with this molecule. This molecule still has some problems, but we changed that in the next scaffold hopping, where we looked through similar molecules similar to these in natural compound libraries and more synthetically accessible libraries. By doing this, we ended up with two compounds that were both synthetically accessible and had high binding affinity, which resulted in us having a substructure, which is an adamantane derivative and a noradamantane derivative. These structures, when combined with the previous structure, which I unfortunately cannot show because of patent reasons, these molecules show high binding affinity against DNA E1, and hopefully we'll be able to provide a drug that is good at fighting tuberculosis. This one?
Yeah, how did you, what was your thought process, what steps did you take? Or was there already a library used, or did you just
So here I can explain that I converted the ligands using the Methyl Python library,
Okay.
But I'm not sure if I can dive into more detail about how this pipeline works, but we will publish this pipeline on GitHub for use for the public soon, but it works by parallelizing the process of docking Vena.
Awesome. Have you tried to replicate your docking data with other docking softwares that are not on Docvina? And also, have you guys ever tried doing trying to replicate this data in vivo to show that it works?
Yes, lab testing is so lab testing is in progress because we still need to synthesize these compounds to be able to test them because they're novel compounds that haven't been synthesized before. But so that's the kind of the next step to this project. But we haven't tested it in other docking softwares because I've entered and it told me to use our Docvina, and we realized that it gave good results, and we kind of decided to go with that.
Did you do an in vitro study after that? Once you have the molecule, did you go through the wet lab stuff, or
Yeah, that's
still
in
pending?
progress. That's in progress.
It's in progress.
Yes.
Any other questions for our speaker? Thank you very much.